everyone. Today we are going to start with a completely new topic, and that is human histology. My name is Professor Dr. Aksh Dubey. I am working in the Department of Anatomy in Swami Rama Himalayan University, Dehradun. The topic human histology is very interesting, and we see there are a couple of very difficult concepts or so much not so covered topics or concepts which are difficult for the student to understand. So we start this topic, we chose this topic to make them as simple as possible. Our standard would be of undergraduate level, and we will go not on the all details but the major and the important details of this. So let's start with our slides. What are the topics we are going to cover in couple of the next videos? The first one is introduction to the histology and its technique, where we will discuss what exactly histology is and what are the important techniques which we use it. Then the epithelium. Then in next video we will cover the connective tissue, then muscular tissue, vessel histology, bone and cartilage histology, gastrointestinal tract histology, the basic one, the nervous tissue histology, genitourinary histology, and the spatial senses histology. So these are the topics which I am going to cover in the couple of the next video. Let's start with the first one, and the first one is introduction to histology and techniques. To understand the histology, let's assume there is an egg here, as you see here in the A, and we divide it into up to the middle, and on the opposite side you see what we visualize into the cross section. So in the middle, in the yellow part, we see it's clear cut a circular almost equal to the maximum size, and then we see covering the whole area, and that is the maximum width covered in this section. But if you see this B1 and you become slight lateral, the section passes through the slide laterally, you see there is a significant change into the presentation, or here we see the area is reduced, the diameter of the center is also reduced. Now if you see this C1 and you go almost laterally, close to the lateralmost area, we see only a periphery, and the central core yellow region is almost out of it. This is the one section, or the vertical section, where we see in anatomy it is named as coronal section, and it is passing through that. Now with this you can assume whatever tissue we take and it is passes through a particular plane, it's very important how the cells are arranged along this plane, and that plane give you the idea of how the cells are arranged. Now you see here, these are the various diagrams or the pictures you see through the microscope, and you cannot give it a one glow. You in the one section you cannot depict it completely. Never be in hurry in prediction in histology. Just take a couple of more sections, analyze them, and then you can analyze the complete structure. So histology is about the series. So if you create this series of sections, then only you can understand the complete structure. Now in the lower part of the slide, you see there is a D, E, and F. The D1 is the horizontal section. You see the another dimension of the same structure of the egg is there. It is rounded outside, and you see the maximum width centrally yellow area is present. But if you go slide below the midpoint, then you see again the periphery is reduced and the central yellow structure is also reduced. And if you go close to the poles, you see almost a reduced periphery, and the yellow is gone, and something blue colored structure which is present somewhere here in this bottom area is now clearly visible. So these are the two sections, one is coronal, another is the horizontal one, and with this two section we may create a 3D structure in our mind how exactly the cells are arranged. As you practice more and more with the histology slide, you clearly getting an idea how the cells are arranged in a particular tissue. So understanding of this plane in the histology is the key feature. It also depends how you are placing the tissue in these sections. Suppose if you are obliquely place the tissue and taking the section, everything would be changed. So whenever we make a block, wax block, we have to be very careful in what orientation we are going to take a section of the tissue, and that give you the idea of the histology. If you keep this thing consciously, then definitely you are making a good slide and your interpretation would be very, very clear. Now, let's assume another idea. This is the idea of a tube. On the left side we see a vertical one, and the right we see the complete structure here. And let's see what are the sections if you are taking at the various places, how they look like. For example, here in this where A is written, if you take a vertical section, then the tube looks like something this. So this much is visible, and through that you can take an idea what could be the dimension of this tube, how the cells are arranged into the wall, what type of cells are there. So this ideation you can take, but this is not the complete picture. On another view like B, C, D, E, F, you are taking the horizontal section at various levels, and for example B, if you are taking above, you do not see or hardly see any lumen inside, but it gives a very good idea about the cell present into the wall. Then you say take a C, you take a section and you see that at this region lumen is clearly visible, but the gland is not round, it is oval in shape at this area. Now in the D, it take two sections clearly closely, one on this side, another, and they are appearing into the histology slide as the round hollow structures, right? Similarly, in the E, if you take a section, you take a big structure and with the lumen, and if you go with the slightly below that where we see the U-shaped loop in upper part, we see the two lumens which are connected with each other with the tissue, though the area is not connected, but if you imagine then these are the connected tubes or the adjacent tube, or they are the not two tubes, they are the one tube which are visible into the two, that because of the curving at this level. So we have to always assume what could be the probable structure of the tissue if you see this section and make them a 3D picture inside your brain, and that comes with a practice. So in histology, never ever comment on a single structure, what exactly the whole structure is all about. We have to take the different section at the various levels, we mix them all, and then only we can create these pictures. In the present era, there are so many softwares are also available which can create the tissue structure in 3 dimension, but an undergraduate medical student in this presentation, we are not going to discuss those structures. So we take the simple slide, build just the basic procedure, and then we make the idea out of it. Now, in this section of a slide, we see there are the blue colors area, let the blue color dots, and this pink color areas, and they are named as H&E staining. This is the first and the most basic staining of the histology, H&E, hematoxylin and eosin staining. And here we see this blue color is of a dye because it is basophilic. That means it's an acidic basic dye, and blue color is a basophilic structure. The base is there, and it is, we always remember that base is always attached to the acid. So it is attached to the DNA structure, deoxyribose nucleic acid. So base and acid makes one staining, and that appears as blue. The another structure is pink colored, and that pink colored structure is the base. It is acidic structure, so it attached with the basic structures, and we see they are this sort of area. So in the pink and the blue color, these are the two colors, and that create various sort of the grading or the coloration which we see in this slide. There is another important thing which is visible here. Here in this structure we see that this is the longitudinal plane, then this is the transverse plane, This is again transverse plane. Here we see this is the oblique plane. So as you go into the histology, you will have an idea how tissues are arranged, because tissues are not in a one tissue is not in a one plane. Sometimes they are the multiple tissues present in a single tissue, or there is a group of tissues or the group of cells inside that which are arranged in different dimensions, and that gives in one section gives the different orientation. So by this all techniques you have a clear-cut idea how the cells are arranged, how tissues are arranged in a big organ and the big tissue, and when we take the slide of these organs, we analyze basically these tissues. So if you keep these basics, you will be master of histological orientation. That will help you a lot in understanding how things are arranged in a one organ. So in this video, we have studied what are the planes present, how these planes could be arranged, how to interpret them, and what is the basic staining. So in the next video, we come with some another topic. Thank you.